Good afternoon, everyone. So for this session, we're going to discuss about erythrocyte metabolism. So last time, we've talked about the primary function of RBCs. Yes, the primary function is to carry oxygen from the lungs to the different parts of the body, particularly to the tissues or cells where oxygen is released. And this is achieved through attachment of oxygen to the hemoglobin molecule. Of course, this picture depicts the process in which the oxygen is coming from the pulmonary alveolus of the lungs, and then they will attach to our hemoglobin molecule. One hemoglobin molecule is capable of binding 4 oxygen molecules. Okay, and then of course that will be transported and delivered to the cells where oxygen are released. Okay, secondary functions of our RBCs is to return your carbon dioxide to the lungs. So once the RBCs are done releasing oxygen, they will also get carbon dioxide from the different parts of the body and return it to the lungs. Okay, and then another function of your RBC is buffering the pH of our blood. So there are processes in our RBC metabolism that requires energy. One function is in the maintenance of our intracellular cationic electrochemical gradients, where 15% of erythrocyte ATP is produced, where 15% of RBC ATP production is consumed. Okay, so when your cell is deprived of ATP, meaning your RB has not generated, even utilized ATP, your RBCs are easy to swell and eventually will be destroyed. So another functions that will require energy, functions of your RBC, you have maintenance of your membrane phospholipids or lipid bilayer. It needs energy. Another maintenance of skeletal protein plasticity, and then also maintenance of functional hemoglobin, the hemoglobin that is located in the inside of our RBC, and then the protection of cell proteins from oxidative denaturation. Of course, our cells, especially our RBC, it is very vulnerable to oxidative injury from oxidative factors. Then, of course, it requires energy in order for the cell proteins to be protected inside. And then initiation and maintenance of glycolysis. Okay, so it is very important to have energy when you start glycolysis, because based on the process of glycolysis, it utilizes 2 ATPs, and then after the entire process of glycolysis, it will yield 2 ATP. Okay, so it is very important to have energy, okay, to initiate and maintain glycolysis process of our RBC. So again, your RBC is a very simple cell. It is non-nucleated. It doesn't have a powerhouse, doesn't have mitochondria and other organelles. Therefore, it is unable to synthesize proteins and lipids. Okay, so in order to sustain its life within 120 days, to allow the RBC to survive up to 120 days, it should have an intact RBC membrane. It should constantly do glycolysis, and it should have nucleotide metabolism. So for the RBC metabolism, energy is stored in these forms, like your ATP, ADP, and AMP. ATP, okay, adenine triphosphate, diphosphate and adenomonophosphate, right? So ATP, ADP, AMP respectively. And then, yes, your glycolysis is very important in order to generate ATP from ADP. Okay, so it is very important nga ATP jud ang ma-generate because it is the functional one. Okay, so ATP is the greatest reservoir of energy in our RBC. So therefore, glycolysis must really occur and happen. So plasma glucose enters your RBC catabolic process through our membrane transport system. So um, our RBCs, okay, cannot produce energy. It relies on glycolysis, so therefore it needs glucose. Okay, so if there is one molecule of glucose, glycolysis will occur, and one molecule of glucose, okay, will yield 2 ATP. Okay, ATP. Okay, after glycolysis niya ang usa ka molecule of glucose, it will yield 2 ATP at the end. So the processes involved in RBC metabolism, you have two main processes. The major process is your anaerobic glycolysis, which is called Embde-Meyerhof pathway. Okay, it is actually 90 to 95% of the entire glycolytic process. And then the other 5 to 10% is your aerobic oxidative glycolysis, your hexose monophosphate shunt. Okay, by the way, your hexose monophosphate shunt is the one that provides a pool of reduced glutathione to combat potential oxidant injury to our RBC. So, as I've said, oxidative factors can injure our RBCs, our cells, especially our RBCs. So we need to have more reduced glutathione so that we can combat these potential oxidant injury that will destroy our cells or even our RBCs. So this is the whole process of glycolysis, where you have 1, 2, 3, 4, one major pathway and then 3 other bypass pathways. But they are very important. These are important. All of these are important to undergo so that the glycolysis process will be successful. Okay. All right. So RBC metabolic pathways, they are essential, they are important for oxygen transport and maintaining physical characteristic of our RBCs. So let's start by discussing first what is Embde-Meyerhof pathway. Again, Embde-Meyerhof pathway is an aerobic method for energy generation that catabolizes glucose to lactate in order to form ATP. So this is the whole process here. If we're going to go back to that picture, this is the Embde-Meyerhof pathway. Okay, so this is the Embde-Meyerhof pathway. So yeah, it generates ATP. Okay, 2 ATPs at the end of it, and it generates 90% of energy needs. And then 90 to 95% of intracellular glucose through free energy diffused, and then RBC, because RBCs have no glycogen. So again, let me remind you of this picture, Embde-Meyerhof pathway, where there will be 2 ATPs that will be used or utilized at first, and then after which there will be 2 ATPs that will be molecule that will be generated. So na-equate na nigamit man. So naka-provide po siya duha. So -2 plus 2 equals 0. And then pagka last diri, in this part here, there will be 2 ATPs that will be generated. So at the end of this whole thing, whole glycolysis, one molecule, okay, undergoes glycolysis, it will yield 2 ATPs. Next, let's go to Lubering-Rappaport shunt. Okay, Lubering-Rappaport shunt is a bypass pathway of Ende-Meyerhof pathway. So, diba the main pathway is the Ende-Meyerhof pathway. So, bypass siya kay naman siya diri sa kilid. Kaya murag ma-appeal siya, but ma-appeal siya during this stage, this phase of Ende-Meyerhof pathway kani siya. Ende-Meyerhof pathway man ni siya, then this one is the Lubering-Rappaport shunt. Important ni siya mo occur so that makaproceed po siya up to the end of the process. Okay, so it is a bypass pathway of Ende-Meyerhof pathway. It allows RBC to regulate oxygen transport during conditions of hypoxia or acid-base imbalance. So the purpose of this pathway, the Lubering-Rappaport shunt, is to allow accumulation of 2,3-DPG. Okay, there will be such time that 2,3-DPG is very essential. Okay, especially when the RBC is already in the process of releasing the oxygen to the cells. Okay, so without 2,3-DPG, okay, the oxygen cannot be let go by or cannot be released by the oxygen. So, like for example, in this picture, 2,3-DPG must be produced, must be accumulated in this part here in order for the hemoglobin to release the oxygen. Otherwise, if wala tong 2,3-DPG, dili niya ma-release ang oxygen. Kay high man ang affinity sa imong oxygen and kaning heme. Okay? Kung walay 2,3-DPG. Kung naay 2,3-DPG, low na ang affinity. So, kung low ang affinity niya, dali na niya ma-release ang oxygen. So, that's the function of 2,3-DPG. It's essential for maintaining normal oxygen tension. So, formation of 2,3-G, it allows low oxygen tension, meaning dili kusog ang kapit, para dili kusog ang kapit ni oxygen sa heme para ma-release ra niya dayon. Okay? Your 2,3-DPG binds when mag-deoxygenation na, meaning magpagawas ng oxygen. So, hemoglobin resists oxygenation by decreasing its oxygen affinity. Okay? So when there are 2,3-DPG that will accumulate to these areas of your RBCs, okay, more oxygen will be delivered to the tissues. So Ende-Meyerhof pathway, your Ende-Meyerhof pathway utilizes 2 molecules of ATP at the very beginning or first steps of Ende-Meyerhof pathway. And then there will be 4 molecules that will be formed during the preceding stages. 2 is formed from this one, ma-form siya dire, okay, during this one. So, kaya ni-utilize man siya, ni-gamit siya, ni-use. So, dire maka-generate, okay. So, -2 plus 2 equals 0. At least, naka-generate, nabawi niya ang iyang giutang na ATP. Okay, but another 2 will be formed from phosphoenol pyruvate to pyruvate in this part here. Okay. So, naay pyruvate kinase dire, the enzyme that will help this one to be converted into pyruvate. Okay. So, without that enzyme called pyruvate kinase, dili pud ka generate og another 2 ATP. Okay. So, again, okay, it requires pyruvate kinase, and after it was successfully converted, then it will yield 2 ATP, okay, for each molecule of glucose that will undergo glycolysis. Okay, So NAD plus and NAD, the reduced form, are very important cofactors of your Erdem-Ehrhoff pathway. Okay, this one. So again, as I've said, nag-utilize of 2 ATP, naka-generate of 2 ATP, plus naka-generate na po og another 2 ATP. Okay? So again, net yield of 2 APT for each molecule of glucose when being processed with glycolysis. So ATP is required for these pumps like sodium potassium ATPase pumps and calcium magnesium ATPase pumps. So it is very important to have this enzyme when you perform these two. Okay, so without pyruvate kinase, this cannot be done. So again, your pyruvate kinase is usually involved in this area. Okay, converting this one to pyruvate. Okay, so there are instances that a patient can have this kind of disease called pyruvate, or disorder rather, pyruvate kinase deficiency. So when there is deficiency of this kind of enzyme in the body, these two pumps will not function. Okay, these two pumps will not function very well. So let's talk about another pathway called methemoglobin reductase pathway. By the way, methemoglobin reductase is an enzyme that reduces your methemoglobin to hemoglobin. Okay? So, what's the difference sa methemoglobin o kanang normal hemoglobin? The normal hemoglobin has a ferrous state, ferrous form of iron. Ang methemoglobin, okay, has a ferric state of iron. So, ferric ang sa methemoglobin, ferrous ang hemoglobin. Si methemoglobin, di siya maayo because it cannot readily bind to oxygen. Okay? So, kung di siya readily bind to oxygen, then magkulang tag oxygen sa atong body. Okay? So, it is very important to maintain our iron in ferrous state. And then, of course, we cannot do away some oxidative factors in the body. So, naagi times nga, ang ferrous na to, mahimong ferric. Kaya na may mga factors in the environment nga makakos nga mo oxidize from ferrous to ferric. Therefore, we need this enzyme para mubalik na siya o ka ferrous state, reduced form. Okay? So, in the absence, kung wala taan nga enzyme, if we don't have methemoglobin reductase in our body, therefore, the hemoglobin, the iron state in our body, in our hemoglobin will become ferric. So, if it is ferric, then it will then be called methemoglobin. So, methemoglobin will accumulate if we don't have this enzyme called methemoglobin reductase. Okay? So, in this pathway, your NADH acts as coenzyme. Like, for example, this one. Okay? Coenzyme na siya. That needs to be reverted back also. Reverted back to its reduced form. So, same here. Importante kay ang reductase. Okay? Methemoglobin reductase to reduce your ferric to ferrous. Okay? And with that, it cannot act alone without this one. Okay? Di siya maging successful without a cofactor, which is your NADH. So, speaking of methemoglobin, it is non-functional form of hemoglobin. So, we don't want to have this kind of hemoglobin form, variant hemoglobin in our body. Kaya nga naman, it cannot bind readily. It cannot bind readily to your oxygen. Okay? Lost oxygen transport capabilities. Of course, because the heme portion cannot combine with the oxygen. Okay? So this happens, methemoglobin happens when there are oxidative factors that oxidizes our ferrous to ferric. So, naay mga factors yun in the environment everywhere we go nga makakos nga ang atong ferrous state sa atong hemoglobin mahimo ma-oxidize siya into ferric. Okay? So, mura na siya nahimong normal, but we really need reductase, methemoglobin reductase para mo revert back to the reduced form, which is the ferrous, which is the healthier one, which is the one that we want for our hemoglobin to have, so that our oxygen can readily bind to our hemoglobin. Now, let's go to hexose monophosphate shunt. It is also called pentose phosphate pathway. Okay, it metabolizes 5 to 10% of glucose, and it is a method which is aerobic. Okay, oxidative or oxidative method of RBC glycolysis. It protects your RBC from oxidative injury because it generates glutathione, and its main purpose is to provide reducing potential for the cell by producing NADPH. So in this part here, this is your phosphogluconate pathway, or the other names niya, tulong names, other names niya. Pwede mo siya tawag phosphogluconate pathway, pentose phosphate pathway, or hexose monophosphate pathway. Okay. So as you can see here, okay, again, the main purpose of this pathway is to provide reducing potential for the cell by producing NADPH. So NADPH is produced, okay, so your NADP through your G6PD enzyme, okay, your NADPH will be produced. So your G6PD, as I said, is a rate limiting enzyme. Okay, it produces NADPH that keeps glutathione reduced. So mura siya domino effect. Kinahanglan tag enzyme at G6PD para makaproduce ta ani. Kaya kani siya maka-help ni siya to keep our glutathione reduced. Okay? Ngano din si glutathione? Glutathione is the one that protects our cells. Okay? Kaya every time na maka-encounter tag mga oxidative factors like your H2O2, okay, hydrogen peroxide, si glutathione ang mo-break down niya into water and oxygen. When we have a lot of this, okay, our RBC is very, very prone for oxidative injury. Okay? So again, okay, G6PD produces NADPH, and together with the reduced glutathione, it provides a main line of defense for RBC oxidative injury. So what are these factors nga makaproduce of hydrogen peroxide? We have oxidant drugs, we have our pollution, we have UV, and we also have infections that can accumulate hydrogen peroxide. Okay? We really need glutathione para i-break down ang hydrogen peroxide if ma-expose mo sulod siya sa itong body. Kaya when we have accumulation of hydrogen peroxide, it destroys the cell proteins. Okay? It is toxic to our cell proteins. Okay? So, have you heard of G6PD deficiency na disorder? So, if G6PD is deficient in the body, therefore, di siya ka-produce ng NADPH, since wala siya ma-produce ng NADPH, di niya ma-keep ang glutathione to the reduced form para i-break down ang hydrogen peroxide. So, walay main line of defense for RBC oxidative injury. So, kung wala kay enzyme na G6PD again, prone kay ka for cell destructions. Okay? Dali ra kay ka magkasakit. Okay? Daghan kay kang hydrogen peroxide, especially if na mga infection na mo-produce ng hydrogen peroxide. So magkadaghan siya sa body kay wala man tay G6PD in the first place nga mo-produce og NADPH nga mo-keep sa atong glutathione nga reduced form. Nya ang reduced form ng glutathione mao man na ang mo breakdown sa hydrogen peroxide which is toxic to our cell proteins. Okay? So G6PD pathophysiology, this is a defect in the hexose monophosphate shunt pathway. It decreases antioxidant GSH, and it is the RBCs. When there is deficiency of G6PD, the RBCs are sensitive to oxidant stresses. Okay, you will notice that there will be inclusions, and these inclusions are known as Heinz bodies. Kung ihunan kung wala kay kung na kay G6PD deficiency or wala ka ng enzyme ng G6PD. So you will notice that you have a lot of Heinz bodies in your RBCs. So of course, if you have Heinz bodies, it will be recognized by the macrophages of your RES nga ilahang i-phagocytize. Okay? So kay inclusion na siya, ipiting siya. So it will be pitted. And i-bite niya ang cell membrane sa RBC nga na Heinz bodies. Then of course, mura na siya bite cell, diba? And then RBC membrane with underlying precipitated hemoglobin, ma-change niya ang form. And then of course, kung ma-change niya ang form, then the RBC is easy to hemolyze. So enzymes of RBC. So of course, I've mentioned this a while ago, G6PD or glucose 6-phosphate dehydrogenase. It is one of the enzymes in your hexose monophosphate shunt. In fact, it is a mediator of that pathway. It fuels the reduction of your NADPH. Therefore, it provides and it produces NADPH kaya siya man ang fuel sa pag-reduce ng NADP to NADPH. And then the NADPH is the one, it is utilized to reduce your glutathione to reduced form. Kasi reduced form glutathione maumay mo breakdown sa ato ang mga accumulated hydrogen peroxide. And hydrogen peroxide is not good for our cell. It destroys our cell. It destroys the proteins of our cell. So we need reduced glutathione. Reduced glutathione is also known as gamma L-glutamyl L-cystenyl glycine or GSH, or it can also be called as gamma glucisgly. Okay, so it is a principal reducing agent of our cell, and it is used to reduce oxidized sulfhydryl groups in hemoglobin and other proteins in the RBC membrane. And this is required to decompose or to dissociate the hydrogen peroxide to become water and oxygen. So iya siyang bungkagon kaya para dili siya maka-destroy sa ato ang cell proteins. Okay? So this protects the cell, especially hemoglobin molecule, from reactive oxygen species. Okay? So katong mga microorganisms, especially itong mga bacteria, okay, nga makaproduce o hydrogen peroxide, kana siya very bad na siya for the cell, especially sa ato ang hemoglobin molecule. Haguba na siya. So in this picture have these free radicals. These are actually the factors that oxidizes our compounds. And then yes, we have the UV light, our ionizing radiation, smoking, air pollution, inflammation, metabolism. So these actually destroys our DNA. Okay, the cell, especially entirely the cell of our the cells. Okay, one of it is your RBC. Okay, So these actually are called free radicals. Okay. So oxidized glutathione. Okay, we have discussed the reduced glutathione. Now let's discuss the oxidized glutathione shortly. So oxidized glutathione is also known as L-glutathione or oxidized form, or L-glutathione oxidized glutathione, or GSSG. Okay, so it must be reduced in order for the hydrogen peroxide to be dissociated and become water and oxygen. So, kinsa may makapareduce ni oxidized form ng glutathione to reduced form ng glutathione. Of course, okay, that is the job of your glutathione reductase. See, glutathione reductase is an enzyme that reduces the oxidized glutathione to reduce glutathione. So lastly, this one. So 2,3-DPG. It also, pwede na siya tawagan o 2,3-DPG, di or bi, parehas na 2, di ba? So 2,3-DPG or 2,3-DPG, it is the most concentrated organophosphate in the RBC. So asa ganin siya nga pathway, it is a product of Rappaport-Luberin shunt. Okay? So it competes with oxygen for hemoglobin. So kani siya, kuan siya, naara po'y good ani to 3-DPG. In the very start, namukuha pa og oxygen si hemoglobin or ang RBC sa lungs. Kinahanglan dili isa mo accumulate si 2,3-DPG. Isa siya mo insert sa imuhang upat ka hymns. Kay para makuha niya ang oxygen from the lungs. So dapat kani si 2,3-DPG, importante ni siya during the time nga i-release na ang oxygen, okay, from hemoglobin to the cells. Okay? So siya ang mo help para ma-deoxygenated ang ato ang hemoglobin. Okay? So it helps by reducing the affinity of your hemoglobin for oxygen. So yang i-reduce ang pagka-attach sa oxygen o sa hemoglobin para ma-release niya o sa'yo na dahil niya i-release ang oxygen sa mga cells. Okay. So I guess that's all for our topic this session. So what I want you to do, this is a seat work since this lecture is only 30 minutes. So I'd like you to submit a narrative discussion of this pathway, the entire glycolytic pathway of your RBC. So of course, I would expect that you would explain systematically the pathway, mentioning the Erythrocyte pathway and this pathway, hexosmonophosphate pathway, and then methemoglobin reductase pathway, and Luberin-Rappaport pathway, down to the last step here. Okay. Also, I want you to mention there, especially when is the time that ATP is being utilized in the first few steps, and where in this process ATP is being generated, ATP is being generated, and then where in this area is another 2 ATP molecules are generated. And then I want you also to explain that at the end of the process, the glycolytic process, okay, there will be 2 ATP molecules per 1 molecule of glucose, okay, that will be generated or will be yielded, okay, after the entire process of glycolysis, okay, per molecule of glucose. Okay, so please submit it. Okay, I would expect you to submit it within today. The deadline time is 12 midnight today. Okay, basta today. Okay, you're going to submit it today. Okay, so thank you so much for your time and for viewing this video. I hope you've learned something and see you next meeting.